That would be helpful.
Well, yeah, I'll put up the PowerPoint. That's not the problem. The problem is not everyone was there for the lecture. And
Right.
so I like to have the recording, but the recording, like I said, I noticed when I stopped it that it didn't work right. And I'm not sure what happened. There's only the start of the lecture. It isn't the lecture, meaning it's only the first minute or two. And I'm not sure what happened, but if ever you see that red dot isn't going, that means I'm not recording. Somebody noticed when I turned it off at the start. I just it turns on automatically, and I'd rather you waited until I turned it on. And so I turned it off, but now it's on. If ever you see it turn off, it'll tell you that. And then you can remind me you need to turn that on. Like I said, if you don't see that light, let me know. I think it's more visible for you guys than it is for me, because when I share my screen, it's not very obvious. Let me share my screen and take a look here. Yeah, you have to look at that little dot right there. Anyways, that's it. It's only a minute or two, 523 kilobytes for the lecture, which it contains nothing of the lecture. So sorry about that. I'll do my best to find a lecture for today, but there may not be a lecture for today. All right, let's go on for the lab, a recording of the lecture today. And what we need to do is talk about lab 2. Blow that up a little today. And then I need to also talk about your infectious disease project. We are now going to discuss it today, and you may begin the infectious disease project. It is due Saturday, May 14th at 11:59 p.m. The point of this infectious disease project is to make you an expert on one infectious disease. I also need to tell you that you should read textbook chapter 3 through the end of the section on compound light microscopy. You need to have that read before beginning labs 3 and 4, because labs 3 and 4 are going to use the microscope. So that's something you should do as, I guess, homework before lab 3. Any question about any of that? All right, if not, I'm going to shut that down because I've got a lot of windows open because we have so much to cover today. Let's begin with the infectious disease research project. I'm going to blow that up so it's a little bigger so I can see it better. About there. So you're going to be doing an infectious disease research project for 100 points. This is a lab project. These points will go towards the lab. In this project, you need to pick an infectious disease and then send it to me. And if I okay it, you can do your infectious disease on that project. Sometimes students pick diseases which are not infectious diseases, and I'll come back and tell you that is not an infectious disease, so no. Sometimes students pick an infectious disease which has more than one cause, like necrotizing fasciitis, the skin-eating bacteria. There's actually 2 bacteria that cause that disease. So you have to pick one of the or the other, meaning I'm not going to let you pick both of them. And then some people want to do it on pneumonia. Well, pneumonia can be caused by many different microbes, both bacterial and fungal, that get in the lungs. It might even be, yeah, it's also caused by viruses. And so I'll tell you, if you want to do a project on pneumonia, pick one agent that causes pneumonia and do your research project on that. Meaning I do not want you to do a research project on many different species of bacteria and many different species of viruses. Any question about any of that? So you need to clear it with me. And the first person who picks a topic, you're going to get it. So email me your topic, and then I will get back to you and tell you whether you can do it or not. It might be a good idea to have a backup in case your topic is chosen. So that way you have something to fall back on. Generally, I can divide up some projects like pneumonia can be caused by Klebsiella pneumoniae. It can be caused by Streptococcus pneumoniae, and I've forgotten some of the other species, but there's even a yeast that can cause it. So there's different agents you can do it on. And what was another one? Influenza. I can sometimes divide that into, you want to do influenza A or into influenza B. Does anybody know, is there another influenza? Human influenza besides A and B? I've forgotten. There might be a.
Isn't there, Professor, is the N1H1 a different strand than A or B?
No, that's one of the A's or B. That's the haplo, not the haplotype, oh, I forget what that's called. The allergen is done by a, that's the antigens. That's the antigenic variation of the virus.
Oh, okay.
But that is a type strain A or strain B. So you can break it into A or B, and you can limit it to H1N1 if you wanted to, but I would not allow other people to do whatever strain that is. Okay, so I sometimes can divide it up. There's one, I'm trying to remember what that disease is. Septic shock can be caused by two different bacteria. I think one is Streptococcus pyogenes and the other is Staphylococcus aureus. So like two people can do it on septic shock. You just pick one different agent.
I have a question.
Yes.
Do you prefer to be emailed at your Clark email or on Canvas?
I've already answered that. I want you to email me at my Canvas. You can email me at Clark, but you have to realize I'm only going to check my email twice a week at the Clark email page.
Okay,
on Canvas, I'll be checking it at least every other day, and often Monday through Friday, I'll be checking it every day. So pick a disease, get my okay, and then perform research on that disease using books, magazine, news media, scientific journals, the internet, especially if you use the internet and a little bit else. If you're using books or magazines or newspapers, make sure you have a reliable source, okay? Because you can put anything on the internet and you can print anything. Like the National Enquirer, I would not accept that as a literature source, okay? But a textbook would be good. A science magazine will be good. Scientific journals will be good. And the internet, if it comes from a .edu or a .gov page, generally is good. If it's a .edu, just make sure it's not a student-made web page. Your written report, meaning it must be typed, single space, about 1 inch margin. I do want you to have 12-point font. As you notice, I wear glasses. If you use a font smaller than 12 point, I'll have a hard time reading it. And you usually would like a larger font anyways, because that means you can write less. You must have a cover information, but I do not want a cover page. Just put your cover information. That'll be the name of the disease, your name, the date, and then the course. Put that above the first page of your text of your article. So it's on the first page of your text. And then the text is the main body of the information. It's not to exceed 5 pages, single-sided, where you describe aspects of the disease. And I'll tell you a little bit more about the text, but the text is going to be worth 60 points, an additional 15 points for grammar, spelling, flow, readability. You should use your own words. You should only not use your own words if you're putting it in direct quotes, and then say where it came from. Whenever you have information either in direct quotes or information that is not common knowledge, you should give an in-text citation. Okay. For example, if you're going to say the number of cases in the United States that have come down with COVID-19, that's not common knowledge. I know that the number of deaths in the United States is over 800,000, but I don't know the total number of cases. So if you give that, that should be common knowledge. And even the number of deaths, even though I know that, and it's something over 800,000, that's not common knowledge. So you should give us in-text citation. And I'll talk a little bit more about citations in a minute. So anything that's not common knowledge or you directly quote, you should have an in-text citation. Now, if ever you have a paragraph and every sentence is not common knowledge, you do not have to give an in-text citation after every sentence. If every sentence in the paragraph have the same source, then you can give at the end of the paragraph the in-text citation. However, if every sentence in that paragraph has a different source, then you have to have an in-text citation after every sentence. Is that clear? If you do not have something that you have in quotes, or you don't put it in quotes and you take it from somebody else, that would be plagiarism. And that will result in a score of 0 on this assignment. So don't plagiarize. In-text citations are worth 10 points. And I'll talk a little bit about how the points are broken down in just a little bit near the end of this, I think. You may use any common citation format, APA, MLA. I think you did MLA in the plagiarism quiz, but it might have been APA. Those are two very common ways. The thing is, just pick a citation format and then always use that format. Don't use one format on the top of the paper and another format on the bottom of the paper. So be consistent throughout the assignment. And this is telling you what sources you can use for citations. I'll let you read about that. Be suspicious of some sites like religious, political sites, privately funded publications. And the problem with these sites is that they have their own viewpoint. And so they may not be sites that would be acceptable. I don't want to say sites that are not telling you the truth, but they may not be telling you the truth. And that is they have their own bias. Okay, maybe a political site I can talk about because nobody will care. I think everybody knows that you're not going to state for something to prove your argument from a political website or a political article, because the article or website has its own bias. That's why we call it a political source. Okay. You should be careful of personal websites because anybody can put up anything on the web. And then college and university pages that have student work, meaning if the student makes the page, it may not be factual. You are not supposed to use Wikipedia because anyone can go into Wikipedia and change it. And I actually saw somebody, another instructor, go into Wikipedia and change something on Charles Darwin just to see, can you go in and change something that they look at all the time, because lots of people go to Charles Darwin and put up all sorts of strange stuff. And he was able to go in there and change it. So the point is, you can't use Wikipedia as a source. You can use it for getting information. You just have to have another source to back it up. And you can scroll down to the sources or references in Wikipedia to get that reference to back it up. Is that clear? So you can't use it as a reference in your paper, but you could use Wikipedia as long as you get an actual reference. You have to have in-text citation and literature citation page, and I'll explain the difference in a little bit. You should use the same citation style. The literature citation page is worth 15 points. And let me warn you that in the in-text citations and the literature citation page, students frequently lose points. So pay attention to these parts of the paper. They may not have the literature citation exactly correct, and so you're going to be missing points there. You must submit the point and the folder to the folder on Canvas, and it will only accept a Word document, a docx document, that's a new Microsoft Word document, a rich text font document, RTF document, or a PDF format. Okay. Any other format, you cannot submit it, and don't go in there and then change the, what do you call that? The subscript of the document, because it won't work. And then you'll get nothing on that project. And I'll tell you that this project isn't working. You can submit it late for partial credit, but don't submit it. Don't do that, and then you'll have to submit it late. I'm just going to word it that way. I've got this in here because sometimes students use something else. Like what's another word editor, not word editor, what do you call that? Text editor. Okay. Nobody's mentioning another text editor. I can't use those other text editors, so I wouldn't be able to open it. I can open docx, rich text format, PDF. So that's why I limited. All right, your text should have a subheading that discusses the causative agent, and it should go over this information. I'll let you go over that. Obviously, it'll depend whether your agent is a bacterium, a virus, or a prion, or a eukaryote, such as a fungus, and get the information you need. Then go over the history of the disease. And I tell you, you should look into how long we've known about the disease. Historically, how was the disease treated? What's the history of the vaccine's development? And if there's no vaccine for the disease, just state that. I describe the changes in our knowledge and attitude towards this disease throughout history, like smallpox, or some other disease that's been with humans for a long time. anyways, strep throat, for example, is a disease where we've changed our knowledge and attitude of this over time. Something like COVID-19, which is not very old, what is it, 2 or 3 years old at most? We've had an increase of knowledge even in that time, and we've changed our attitudes a little bit. Okay, although it's less lethal now, and there's people who have been vaccinated, and so many people have been infected that the disease doesn't spread the way it did initially. So that's a reason for a change in the attitude. So even with a new disease, we've had a little bit of change in knowledge and attitude. Go ahead and talk about the epidemiology, the pathology of the disease, the response and treatment, and then the social and political economic considerations. So each of these topics will be worth 10 points. With the exception of the causative agent, it's 9 points because 1 point goes for the introductory information, the heading information. And here it'll be graded 1 point for the cover information, 9 points for the causative agent, 10 points for the history, epidemiology, pathology, response and treatment, and then social and political and economic issues. You'll get 15 points for the quality of your writing, 10 points for the in-text citation. And let me state how you do an in-text citation. And then the literature citation page. The literature citation page should have every in-text citation you listed, but instead of being an in-text citation, it should be the full reference that should generally have the author, the year, the source of the publication, and that's usually the article name, and then if it's a journal, the journal name. If it's a web page, you would say the article name and then the web page address. If it's a book, you give the article name, and that could be like chapter 2, whatever, chemistry. And then you give the name of the textbook, and the publisher of the textbook, and the city of publication. So the literature citation page will differ for where your source of your article comes from. Okay. You must have 4 reputable sources. And for the literature citation page, I will tell you this, it should have been in your plagiarism information, that you are not allowed to give an abbreviation in the full citations, meaning you have to give the full names, last names at least, and initials for all of the authors, unless it's something like 7 or 8 authors. I'll have to look it up. I don't remember. 7 or 8, I remember that point. In which case you can then go ... and then give the last author. So you give something like 6 or 7 author names, and then you go ... You don't go any further. And then you give the last author's name. So generally, it's best to give all of the author's names for the full text. And then it must follow whichever citation style you chose. Like I said, for a web page, you should have the author's name and the year, if you can find it. If then the article name, and then the web page address. 4 reputable sources, and I talked a little bit about what's a reputable source up here. Where was that? There it is. Generally, something from a government page is okay. Scientific journals, Nature Science, popular science magazines. Generally, textbooks are very good. You'll note that all of your textbooks are not referenced, what's that called, where it's reviewed, okay? If it's a credible newspaper source, you can take it, like the New York Times. But like I said, don't take anything from the National Enquirer. All right. Let me talk about an in-text citation. Let me just start a new page here. For an in-text citation, what you do is you have your text. We're just going to go like that. And then you go in parentheses, and then the author's last name, comma, and then the year. And then parentheses, that's the in-text citation. All right. Now, if it's two authors, I'll warn you that you should go author one. Well, let's go ahead and put the parentheses in. Author one and author two, their last names, and then the year. If it's more than two, you go author one. Oops, that shouldn't be that way. Author one, semicolon. I'm drawing a blank here. Sorry, no semicolon. Et al. And that shouldn't be a capital A, comma, and then the year. Any question about that? All right. That should be all in the plagiarism information. All right, that's it for the infectious disease project. Any questions?
Are we
I have a
emailing? Oh, sorry. Are we
Sorry.
emailing you our topics, or how do you want us to submit that?
Yeah, send me the topic, especially in Canvas. You can do it to the clark.edu, but realize I won't be answering that right away. So best to do it in Canvas. And let's see, somebody else had a question?
Yeah, when you send it in Canvas, like that's just using the inbox feature, right?
Yeah, that's the Canvas
I
email, right there. See it?
Yes, I sent you my topic last Thursday in between lecture and lab, and I haven't had a response yet. So I was just wondering.
It's because I hadn't talked about it yet. I'll take a look at it now. All right, let's move on. We need to talk about the lab. Lab 2, aseptic transfers. Upon successful completion of the lab, students should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data provided, and be able to define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. In our local environment, microorganisms are ubiquitous. That means you can find them almost everywhere. This is a problem when working in microbiology because we generally want to work with pure cultures, a culture that contains one and only one species. Louis Pasteur developed the technique of making a pure culture and then using aseptic transfers to minimize the risk of culture contamination. And we'll learn how to make aseptic transfers in this lab. In a future lab, we'll talk a little bit about how to derive a pure culture. Any questions about any of that? All right, so inoculation is moving cells from one area to another. And we usually have a stock culture and we'll use it to inoculate a tube or a petri dish. And that we call inoculation. During inoculation, never lay the tube cap down on the counter or workbench because it can get contaminated. Always hold it in your hand. And to minimize contamination when inoculating the media, let me see if I can show this. I don't have anything empty, so it's hard to do it. I'll use this and hopefully, can you guys see this? Well, I'm going to assume you can see it because nobody's answering. If you can see this, you want to hold it at an angle and then insert your tool in the angle. The reason is if it's at an angle, if something comes down from the air, it may not go in the inside the tube. Whereas if you hold it directly up and something falls down from the air, it's going to go right in the tube. So hold it at an angle, put your tools in there and do your stuff, and that will reduce the risk of contamination. For a Petri dish, what you should do is never take the lid off and then put it on the counter. But when you're going to inoculate a Petri dish, take off the lid and then use the lid to partially cover. I don't know if you can see this when I put it down. Can you guys see that?
No.
Let's bring it down. Can you see it now?
Yep.
Okay, great. You want to hold it so that you're partially covering the tube. And I really need a pin here. I don't have one, so I'll just use these glasses. And then use your tool to inoculate the plate. And that way, if something were to fall from the air, most of the time it's going to hit the plate and it won't go into your Petri dish. Obviously, if something falls right here and something happens to fall right here, you're going to get contamination. But you can reduce it by angling the tube or keeping the plate partially covered. You should work efficiently, not talk while you're doing it. When you feel a cough or a sneeze coming on, cover the dish, okay? All right. In order to inoculate a new sterile media, that's transfer cells into the new media, you must make sure that you sterilize everything that can potentially contaminate the sterile media. Generally, the media given to you will be sterile, put in autoclave and then cooled and then given to you. And then what you do is you sterilize your tool, like put the loop into the Bacto incinerator and get the wire portion of the loop red hot. That'll sterilize the wire portion. And then only insert the loop portion into the tube. Any question about any of that? All right, know the different terms. In laboratory 2, you're not physically going to be performing any of these activities, but you will be watching videos on the activities. We will expect you to know and understand the material in the videos. For procedure 1, you're going to transfer a sterile liquid using a glass serological pipette. In procedure 2, you're going to transfer a sterile paper disc using forceps, which we'll call semi-sterile. For procedures 1 and 2, please watch the video demonstrating proper aseptic transfer for a serological pipette and presumably a forceps. I guess I should watch that myself because I don't think I've done that. In procedure 1, you're going to aseptically transfer 1 milliliter of sterile water into a nutrient broth. So I got a picture down here. This is the nutrient broth, and you're going to put 1 milliliter of water in a pipette, and then pipette that sterile water into the media. Any questions about that? When you're using the pipette, you should note that the water will form a meniscus, on any glass or plastic pipette. It'll be worse on glass, but you read the meniscus from the bottom of the meniscus, not from the top of the meniscus, and not, I don't know, in between the top and the bottom. All right, for procedure 2, you're going to use aseptic transfer to transfer a sterile paper disc into the nutrient broth. So we'll give you a paper disc, and using forceps, you put that into the sterile tube, and you do clean the forceps before you do that. Okay, so that's procedure 2. Procedure 3, you're going to be transferring Serratia marcescens into a broth. A broth is a liquid culture. So we can use this tube here, that's a liquid culture. And what you do is you get some cells on a loop and then inoculate the liquid culture. So that's procedure 3. And then procedure 4, you're going to transfer Serratia marcescens onto an auger slant. An auger slant is a solid culture. There it is right here, a solid culture. And you get your bacteria onto a sterile loop, Serratia marcescens, and then put it on the surface of the auger slant. I'll talk a little bit more how you do that. For procedures 3 and 4, please watch the following video demonstrating how to correctly transfer from a bacterial stock culture to a sterile broth and to a sterile auger slant. A note about these videos, we're going to be using Serratia marcescens, which are reddish cells, so they're pigmented. In the video, I think they're using E. coli, and it's non-pigmented. I think this is the first question. There's actually an error made when the instructor puts the loop into the E. coli stock tube. See around minutes 3 to minute 3:20. Did you know what the error is? Hint, you can hear sizzling when he puts the loop into the E. coli. All right, so talking about procedure 3, using aseptic technique, transfer Serratia marcescens from one tube, which is the stock culture, to sterile broth. You should note that Serratia marcescens, when it's grown on a solid, will look red, and when it's grown in a liquid culture, the liquid will look pink because it's a pigmented bacterium. A broth culture is problematic because it is difficult to determine if contamination has occurred because all the cells will mix together in the liquid broth. And so if there's minor contamination, you wouldn't see it in the liquid broth. But if there's major contamination, you would probably see it because the broth would not look pink. Any question about any of that?
I have a question about the forcep. You said it was semi-sterile and we would need to clean it beforehand. What are we cleaning it with?
I think they state that in the directions here. It doesn't tell you how they sterilize the forceps, which, let me see. An easy way to sterilize the forceps, and it gets them relatively clean, but not really sterile, meaning sterile has no microbes on it. you can put the forceps, clean forceps, obviously, in alcohol and then flame the alcohol. And that gets all of the vegetative microorganisms killed, but that will not kill an endospore. Okay. Another thing you can do with the forceps is to put it into the flame. I think we got a picture of that. Put it into the flame or into the Bacto incinerator, but you can't get it totally red hot because you have to be holding it. Okay, so you just briefly put it into the flame or the Bacto incinerator and then let it cool. So that's how you sterilize it. And this lab doesn't really talk about that. So don't worry about it. Okay. All right. All the tubes should be appropriately labeled. I'll let you read what you should label them with. In procedure 4, you're using aseptic technique to transfer the Serratia marcescens to the sterile auger slant. On an auger slant, you can get high cell densities that are grown, and contamination can be easily determined because on your streak with Serratia marcescens, you should have a red streak wherever you streaked it, which would be this area here. And if ever you have a region which isn't red, then you know that's contamination. So you can see contamination on a slant. You cannot always see contamination on a broth. So if you're in the lab, you'd be making these observations and doing the experiment. But in the lab, on an online class, we're going to give you the table, and you should be familiar with the possible outcomes and the associated terminology given in this table. So in procedure 1 and 2, if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer is successful. So aseptic transfer is successful is your conclusion. In procedure 1 and 2, the result is if nutrient broth is turbid, hazy, or cloudy, the organisms are growing in the broth and aseptic transfer was unsuccessful. That is clear, and that of course is uninoculated, but if it were inoculated and it would be clear by that, then you would know that nothing is growing in this tube. Any questions about any of that? And then if it's hazy or turbid, you will see haziness or turbidity in this tube. Any questions? For procedure 3, if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was unsuccessful. Because remember, we are purposely transferring the Serratia marcescens into the liquid broth. In procedure 3, if the nutrient broth is turbid, hazy, or cloudy, organisms are growing, and aseptic transfer was successful. For result 4, if the slant does not show confluent growth, or there's no colonies, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show confluent growth or colonies, then the organisms are growing on the slant and aseptic transfer is successful. For procedure 1, use this tube here. For procedure 2, use this tube here, and you can actually see the paper down there. For procedure 3, use this tube here. And for procedure 4, use this tube here. Any question about that? These are the results for procedure 1, 2, 3, and 4. After the student has done the what do you call it? The transfer, okay? And we're giving you the pictures because you can't do the transfer online. All right, the first question, which isn't labeled here. Why isn't that labeled? Oh, this is my tutors or teacher's version. That's why it's not labeled. Your version will be labeled here. It might be question 0. And it asks, describe the initial clarity of the nutrient broth prior to inoculating. So state whether it's clear or turbid. And then result for P1, describe the clarity of the nutrient broth after inoculating the tube with sterile water and overnight incubation at 35. So state whether it's clear or turbid. And then on question 1B, based on the results, what conclusion can be drawn? So list the conclusion. And to get that conclusion, look in the table right here. And then list 3 sources of error where aseptic transfer technique could explain why the nutrient broth showed the result that it did, the growth of the microbe. So list 3 possible errors. For result 2, describe the clarity of the nutrient broth after inoculating with a sterile paper disc and overnight incubation. State whether it's clear or turbid. State the conclusions. On question 3, describe the clarity of the nutrient broth after inoculating with Serratia marsans and an overnight incubation at 30 degrees. State whether clear or turbid. Based on these results, what conclusion can be drawn? And how do you know whether you transferred the correct microbe? Serratia marsans. Okay. Anyone know how you can tell whether you transferred the correct microbe?
By the color?
Yeah, by the color. For result P4, procedure 4, is there bacteria on the growth of the slant? State yes or no. How do you know? Just give a reason why you know it's yes or no. And then answer, does it appear to be the bacterium you inoculated or a contaminant? You can ignore the references and there you go. Any questions? If there's no questions, go ahead and work on the lab. I will be here until 8 o'clock to answer any question. The recording there, I'm recording this. Go ahead and ask your question now.
So we have from 8 PM tonight to 8 PM tomorrow night to finish the essay question. However, all the other questions have to be done within a time period of when you start.
Yes.
And that
Okay.
time period is how long?
It'll be about 1 minute for each question. Oh gosh, it's almost 8 PM. I gotta get to the quiz.
The second quiz is 13 minutes long, the one with the multiple
Yeah,
choice.
that's not correct. I didn't change the practice quiz. I changed quiz 1, but I need to change the time on quiz 1. At least for the multiple choice quiz. Okay, any other questions?
On the one on the quiz with the lockdown browser, sorry. On the quiz with the lockdown browser, are we allowed to use notes, or is that the purpose of the quiz, is to not use notes?
You can use notes in your textbook, but I'm gonna warn you that it takes you time to look up that information.
Right.
And you won't have enough time to do that more than once or twice. Because if you spend all your time looking up questions, you won't finish all the questions. And
Right.
then you'll run out of time and the computer will mark those wrong.
Okay,
So
and
you
then my
don't
last,
wanna spend much time looking up questions.
that makes sense. My last question is for the essay questions, is there like exactly like how many sentences they're supposed to be, how long they're supposed to be, or is it just kind of depends per question?
It depends on the student and on the question,
Okay,
okay?
How many essay questions is there?
On the quizzes, I think there's 8 essay questions. Except maybe the practice quiz, that might have 10. I don't remember. Yeah, it does have 10, because I think it's 1.1 point per quiz question for the extra credit quiz. But all the other quizzes, I think there will be 8 essay questions. Okay, any other questions?